welcome and thank you for joining us this morning. The ChUG Cytometry group is available. You can access us and follow along with us on our website, which is chugcytometry.com.org, chugcytometry.com. And there we want to point out that we have a forum, a growing forum where you can ask questions and participate in discussions. So when you go to the website, go ahead and create yourself an account, and then you'll be able to say, join one of the forums and ask a question and participate in the discussion there. Think of it as something similar to the former Google Plus cytometry forum that was popular back in the day before Google shut it down. While you're watching things on YouTube, feel free to ask a question in the comment box. We'll have moderators on that live stream who will field those questions and feed it to us, and we will be able to ask Tim either during the presentation or we'll save them for the end as part of our Q&A. So we're going to allow the presentation to go on 45-ish minutes. We'll see how that goes. And then we'll leave some time at the end for Q&A. So feel free to ask those questions throughout, and we'll keep tabs on them and enter them into our Google Doc that we'll be able to bring up those questions at the end. Okay, so today I'm happy to have our first seminar here for the reboot of ChUG. Which we will be talking on the dimensionality reduction and clustering tools for high parameter discovery. Myself, my name is Ryan Duggan. I'm a senior scientist at Abbvie in North Chicago. I'll be one of your moderators along with Laura Johnston, who is the director of the flow cytometry core at the University of Chicago. Monitoring things for us on YouTube will be David Leclerc, who is the technical director at UChicago, their flow facility. And I'll introduce our speaker today, who is Timothy Quinn. Sorry, I always do that to you. Timothy Crawford, middle name Quinn, correct? Tim is, as many of you are familiar with, he's a holdover from FlowJo, so you know him from the FlowJo days, now part of BD Bioinformatics. He's a PhD scientist and FlowJo guru. He goes around the country and gives many of these presentations to lots of different groups. So we're happy to have him here with us today. And again, Tim is going to go through his slides, and as needed, we'll go ahead and interject with questions as we deem fit. So with that, I will turn things over to Tim. Tim, you can go ahead and share your screen and your slides. And
sounds good.
If we can just get, David, if we just can get a confirmation that things are showing up on YouTube properly as expected, we will go ahead and start the presentation.
Well, I have confirmation from a few of my colleagues that
Excellent.
It is streaming online, so we should be good to go. I'll wait for David. Get started. As Ryan said, thank you, Ryan, for the introduction. My name is Tim Crawford. I'm a field application scientist for the central US and Canada with BD. And FlowJo was purchased by BD several years ago. And so we become the informatics group for BD. My email in the bottom left-hand corner, timothy.crawford@bd.com. If you'd like to ask questions or get a hold of me for any reason, please feel free to reach out. Am I good to go?
You are good to go, Tim.
Okay, all right. So we're going to be covering a lot of material in a very short period of time. Sometimes I have up to 2-3 hours to give this type of presentation, so I'll go fairly quickly. But what I really want to focus you guys on is the workflow required for discovering complex populations from high parameter data. And so as a synopsis slide here, I'm going to show you this workflow, and we're going to go through each step describing the different tools that are available within the FlowJo platform, how to utilize them in this workflow so that we can answer the question of really what are the complex populations in a high parameter data set, and how do they distribute across different experimental conditions in your assay. So the first thing we're going to do, of course, is clean up our data. That's going to be standard gating, along with potentially some algorithms to clean up any of the noise or spurious events that come out with clogs and other anomalies from your instrument. And then we're going to label the individual data files and add keyword metadata, which are actual sample level descriptive terms that allow you to describe what where these samples came from, how they were processed, and essentially use that keyword data when we merge samples together to create new parameters that allow us to pull apart the individual samples from different conditions. In this case, I've got different tissues and different time points. And so as long as I correctly label with the keywords, when I concatenate and merge samples together, I get these new parameters that allow me to pull apart the individual component samples from the total. Now, this is important because you can't run dimensionality reduction the way you think of gating on a sample. You can't run dimensionality reduction on a single sample one at a time because these algorithms are non-deterministic. And the output of a dimensionality reduction process is a new graph, 2 new parameters that redistribute events into a 2-dimensional space. If you run dimensionality reduction on each individual sample, you'll get a different output plot every single time, so they're no longer comparable. But if you merge those samples together with the concatenate function, then dimensionally reduce and cluster on that total set of events, you get a common dimensionally reduced data space within which you can explore and observe changes and differences between the different conditions. So after we dimensionally reduce, we're going to cluster, and actually clustering and dimensionality reduction are interchangeable within this workflow. Going to be different phenotypic populations, and it gives you a visual space on which you can overlay clusters and view their distributions. Once we have those cluster populations, we can explore their phenotypes, identify what their expression patterns are, and we can gate apart the different compartments of the different conditions in our assay and view the contributed events within this dimensionally reduced data space, and use these gates, knowing what we put into the system. We can use these gates to understand how each of the individual clusters is distributed and identify the cluster populations that are upregulated or downregulated under the different tissues or the different time points between sick and healthy, whatever the experiment is that you're running. And once we have these populations, we can actually go one step further and dig deeper into them. And I haven't showed on this slide, but we'll be able to generate computationally derived sort gates for a specific population, allow to sort relation that we found with a high parameter dimensionality reduction and clustering algorithm approach, give us a linear or hierarchical gating structure to isolate that population on a sorter, and then we can put it into a single cell RNA-seq or a multi-omics experiment, ask more questions about its expression pattern, both at the protein and the RNA levels. And so how do we actually do these steps? Most of these algorithms are available within the FlowJo platform as plugins, and plugins are just applications that extend the functionality of our FlowJo package. You can get these applications from the FlowJo Exchange website. You can link to it from the FlowJo website, and you'll find a plugins menu within the workspace tab of your FlowJo workspace. And initially it'll be blank because you have to download these plugins and install them to use them. And if you click on the FlowJo Exchange link, it's the first line item within that plugins menu, it'll take you to the Exchange where you can download the actual code that runs all of these tools. So you can break down these plugins into several different steps in the workflow. Pre-processing plugins, you might do something to data upstream to clean it up or to normalize samples across multiple runs. The dimensionality reduction algorithms like TSonique, UMap, and TriMap, they create new parameters, redistributing events into this new dimensionally reduced data space. It's called embedding the data in a lower dimensional space. And clustering algorithms will create populations, just like you gated for a population. The output is actual gated populations, but they're complex populations without a hierarchy to. Ultimately, when we put this together, we can spin up our Cluster Explorer module, which allows us to view the clusters within a dimensionally reduced data space. And then we can spin up our hyperfine algorithm to generate the hierarchical gating to sort those populations and look at them in a deeper or a different assay. Now, when we look at pre-processing, I just want to make you aware of these plugins that are dependent on R, Saitonorm, FlowAI, and FlowClean. As an aside, some of these algorithms are actually interacting with another computer environment, the R statistical computing environment. So those algorithms in bold italic do require the R system, and you will have to do a little extra setup to make that work. But we have documentation that comes down with each of the plugins when you download, showing you what you need to do to set up R, along with the script to install the package and the algorithm within your R environment on your computer. Now, if we want to clean up the data, of course, we're going to gate down to some population of interest, but we worse clogging issues as they were run through an HTS high throughput system on my instrument. And you can see in the bottom row, the FlowAI events were removed. In the black events are what FlowAI actually removed. So it's cutting out the bad data and then stitching back together the good data. And what you can do is run this algorithm and then use just the good events downstream for all your other processes and effectively clean up the data. The original observation in the FlowClean paper was that 13% of public repository FCS files have some sort of fluorescence anomaly and should have been cleaned up.
Hey, Tim.
So it's just, yes, Ryan.
Sorry. Yeah, you were cutting out a little bit, but you know, speaking of cutting,
Yeah.
so have you kept up with the recent discussion on the Purdue list about FlowCut as another one of these cleanup tools?
A little bit, maybe not in the
guys,
past 2 days, but we haven't implemented FlowCut as an option yet.
But
and I don't know what the status would be of that, but
okay. But any idea about like how it performs in terms of FlowAI and FlowClean, and whether, because I think some people say that FlowAI is a little bit too aggressive.
Aggressive, yeah. That has been the observation, and FlowClean may be a little not aggressive enough.
Okay.
And FlowCut is another option which we may consider putting into a plugin, but I don't know the status of that. I have only done direct comparisons with the FlowAI and FlowClean algorithms on my own data. And I promote FlowAI because it does a good job on my Fortessa data from my time as a bench scientist of cutting out these little pulses in and pressure issues that you may see with samples that have poor viability on your HTS unit. And so in my own experience, I've only tested the two, and I prefer FlowAI generally because it does a good job on my own data, but I don't know about FlowCut, unfortunately.
Yeah, I would agree. I think I've used FlowAI pretty much since it came out and have been happy with it. I know somebody else had mentioned that there was some issues with cytometers that were specifically using peristaltic pumps, and that FlowAI was picking up the subtle undulations in the peristaltic pump, and so there was but apparently there's ways to fine tune it a little bit.
There are ways to fine tune it. I'm not going to go into that much detail right here, but we have a great group at my office, the tech support group led by Ian Taylor there. Will definitely help you dive into those questions if you have a specific question about how to do that. And I can follow up on that question with my bioinformatics team and get recommendations in how to tweak some of these options to make it a little less sensitive so it could be used for that type of data set.
Great, thanks.
You're welcome. Thank you. Now, Saitonorm is something that we have implemented just in the past couple months. It's an algorithm from Sophie Van Gassen's group in Ghent and Nina Ajipour at Stanford. And this is an attempt at normalizing across multiple batches, because as good as we are at the bench as scientists to process identically our data sets through the thawing, staining, and acquisition of the data, there is still going to be batch effects. And what this algorithm does is take your raw data and it runs a FlowSum clustering algorithm and then produces metrics across the different control samples. So it's the same control that you have to run on each batch run, each assay that you process. And it then normalizes those control samples, forms a gradient that allows it to shift and corrects for all of the data to normalize it across these different runs. So the first run had lower expression, the next one, the third run had higher expression, and it normalizes to the middle of the two. It then goes back and maps the other files to the same FlowSum map and then adjusts each metacluster population specifically. So it's a population specific adjustment instead of a global adjustment. And I bring this up because most plugins you actually initiate on data within FlowJo by pointing to a gated population or a sample, and then going to your plugins menu and firing off the plugin from the menu downstream. This particular plugin is implemented as a workspace plugin. So you go to plugins and add the workspace plugin, choose Saitonorm. There's only two options. And the output is actually new files. And so what you do is you put your raw files in here, and then you fire off Saitonorm. You specify which samples are controls and give 1, 2, 3, 4 for the different batches to assign different samples to the runs, and then click OK. And the output is a new set of normalized files. So you just run this as a standalone plugin in a workspace, and it spits out new normalized files into a folder, and then you load those separately into a new workspace and work with them downstream. So it's a little bit different from the other algorithms that we have as plugins. And I bring it up because so many people are trying to do longitudinal studies or have clinical samples that are coming in just a few at a time, and you want to collate all of that data together. And there are over time batch effects that make it more difficult to merge all of that data and analyze it as one single experiment. And this is the first attempt at correcting for those batch effects that has been implemented in the R environment and that we've plugged into FlowJo so you can use it and test its utility. Now, however you clean up your data, you're going to end up with some gating subset of populations. And I'm going to, of course, promote cleaning out your doublets and your dead cells and getting down to some population of interest. But then you're going to have a bunch of different samples with different numbers of events in your gated populations. And one way we can normalize the number of events going into an algorithmic process is to downsample those parent populations so that they have the same number of events going.
I think you're frozen
across
there,
the
Tim.
entire selection there. Oh, I was frozen.
You froze for a little bit. So if there's something important you said.
Okay, well, I will repeat it a little bit slower, and we will see. So downsample is point to a population, fire off the plugin, tell it how many events and what the new population name is going to be, and then you get an output that is a distinct population with that number of events. It's a child of the parent. Now you can actually batch any of these downsample operations. This is the downsample node that actually performs the operation. You can batch it across a group of samples by applying it to a group-owned gating tree, and that will fire off across all the samples in your group, giving you equivalent populations that then can be used downstream. And it's about as simple a plugin as it gets, right? Point to the population, fire it off, put some metrics in there, and click OK, and you get an output is a new population. Now, the reason why I want to normalize, as I've mentioned before, is that we want to merge these samples together. And it seems logical to me that I want to balance the different contributed numbers of events between samples and between experimental conditions. So if I have a simple stim experiment, for example, and I have two patients that are either stimmed or unstimmed with something, I can now merge those samples together. And when I do merge, I will always get a sample ID parameter, which places those samples in the order of the workspace list when I initially did the concatenation. So whatever sort order in the FlowJo workspace you started with, those samples will be distributed across the sample ID parameter. But I can also numerically give numerical values, ones for the first two samples and twos for the latter two samples, as a keyword. And I'll make my keyword as a column header in my workspace called experimental condition, for example, give ones for the first two, twos for the next two. And then when you create the concatenated file, you can specify this keyword to be created as a new additional parameter. And it will now merge all the events that are labeled with a 1 from the original samples into a single population along this new derived parameter. So ones around 1K and twos end up as 2K. Now I can split these two populations just by gating across experimental condition for unstimulated or stimulated. And it doesn't matter whether I have 2 original samples or 30 or 100, they all end up at the same position, making it easy for me to gate apart these conditions. Now, if you've never been to one of my presentations and you're like, what are keywords? Well, keywords, again, are metadata, descriptive terms at the sample level that you can add within FlowJo. You can also create them on the front end when you acquire the data. But if you want to add them in FlowJo, you go to your workspace tab, add a keyword, give it a new name, and then you can fill in the column that appears with the appropriate numerical values to order and sort your samples in the concatenated file the way you want to. So I'll take all of these files. In this experiment, I've got 16 files, 4 different time points, 4 different tissues from the same individual, and I will concatenate them. And I'm representing in orange the downsampled events, the same equivalent numbers of events from each file that are now going into my concatenation step. And when I concatenate, I can use these keyword labels for tissue and time point to create the new additional parameters. Now, the details there on the concatenate graphical user interface, right? The window that pops up when you concatenate. Initially, you will select multiple gated populations and you want to merge those populations together. So if you right click, there's an export concatenate function in the right click menu, and it'll initially say export with this tab selected, and the status of the operation is shown down below. If you have multiple samples and selected and you're exporting them, you'll get multiple new data files. But if you click the concatenate tab to the right here, then it merges those samples into a single new data file when you press concatenate. You want to write an FCS3 format file because you're bringing it back into FlowJo, and the destination is the location of the save where the file will happen. Sometimes you want to include all events. That would be appropriate if you downsampled using the downsample plugin, or if you just wanted to include every event and not worry about the normalization of numbers of events. But you can also check this box to include no more than. It will be populated with a grayed out number that is the fewest events in all of the gated populations that you've selected. But this include no more than is effectively a downsample operation during the concatenation step. So it only takes that number of events from each file and merges them. So you can do the downsample right here in the concatenate operation if you like. Generally, we're going to write uncompensated parameters because you want the raw data plus the compensation matrix that came with that data. And this is appropriate if your experiment is all done in one experiment. If you do have multiple runs of data, you may want to write the compensated parameters, which will only write compensated numbers, but it'll be correcting the numbers with the matrix, the compensation matrix from each of the individual experiments. And then you get compensated data downstream, and you don't have a matrix associated with it, but you can work with those merged samples as if they were all the same experiment now. You have some naming options. Concat is the prefix that shows up, and I always give my concatenated files some unique suffix to describe what I'm looking at. Generally, you're going to concatenate all the files together, and then this is the little box in the bottom right-hand corner for additional parameters where you can choose the keywords that you want to create as these new parameters to distribute your individual samples across some condition that you want to compare. When you click concatenate, writes the file to disk, you can choose to load it into a new or existing workspace by checking the box, and when you close the window, it'll bring it back in. You double click on that sample, look at the additional parameters that you've created, and you should see your events distributed appropriately at these different 1K, 2K, 3K, 4K marks, depending on the integer values that you gave them in the keyword columns of the original files. So that's the file we're actually going to work with. And
Tim,
downstream, we can now dimensionally reduce and cluster that.
Tim,
Yes,
before you
Laura.
move on, can you just quickly explain your little trick for using the asterisks for your?
Sure. Well, asterisks here, my keyword, I always put an asterisk because I learned FlowJo back in the Mac FlowJo version 8 days. And when I first learned FlowJo, one of my colleagues showed me that if I create keywords and give them an asterisk, then my own user entered keywords, they show up in any list of keyword options all together. They group together because of the asterisk, as opposed to being in an alphabetized list in different places. And so that is my trick. I always put an asterisk at the beginning of my keyword name for the column. And then in many different areas, when you display keywords or assign them to be created as a part of a table, you will have a quick way to search for and access all of your user entered information, as opposed to all of the other keyword information. And there's a lot different fields that are already there by default in the FCS standard. There's a bunch of $signs, and then there's a bunch of alphabetical keywords that are added by each individual manufacturer. Does that make sense? So it just groups together in my searchable keyword list, so I can find these keywords and effectively get them and use them downstream.
Yeah, I like using that trick. Very useful.
And when I have a live demo, I show that to people, look how easy it is for me to search through my keywords and access them with an asterisk. But I'm not doing a live demo here today, just not enough time for it. So we end up with this concatenated file. And I want to reinforce that this is one of the most important steps, right? I don't want you to scale up and start exploring a file unless you know you can pull it apart effectively and ask that question of how do these clusters change across the conditions in my assay. So, making the concatenated file and being sure that you can pull it apart in the ways you want to compare the different samples is essential before you scale up to millions of events and do the dimensionality reduction and clustering steps, because those are the more computationally challenging steps that will take a longer period of time on your computer to run. And when you run the dimensionality reduction and you don't have the clustering right, well, then you start off back at the concatenate step to remake your file and go through it again. So I say downsample to a limited number of events for your first tries at this, and before you scale it up to millions of events, because then you're going to be waiting a long time for these steps. So the concatenated file allows us to, again, dimensionally reduce, to create new parameters, and then create clusters using a computational algorithm. The dimensionality reduction space that you create, this TSonique space in this case, can be effectively explored with this third parameter color map option that's available in the FlowJo graph window. It's a checkbox, and you use the third parameter to heat map across this data space. And so it's really easy to take your raw TSonique plot, turn on the color map, and just scroll through the different markers in your panel, and you'll find that these markers are expressed regionally, and more redshifted means higher expression in that area. And you'll easily be able to pick out the different lineages, and then some of the other markers downstream of those lineages that are expressed in gradients across the different continents, such as 45RO, which will, of course, determine memory state in the T cell compartments, and others. And so use that third parameter color map to orient you with the different regions or islands of this continent as to what they actually do. And then we can gate apart. Again, we're going to gate apart the tissues and apply those gates downstream of any of these clusters or other gated populations that we want to explore. And we know that we start out with the same number of events from each condition, which means I have this 25% across the board. And if I apply those to any downstream population, then I'll know what the distribution is. If there's no change across these different tissues, well, then the distribution for a cluster would be 25%. But if blood has higher levels than others, then it's enriched in the blood. So we can also gate within this region, this data space that we've created. And this is an effective way to explore the data space. Even before I had color mapping or any clustering tools, I could draw a query gate, which is just a gate within the TSonique or dimensionally reduced data space, and then make an overlay in the layout editor of FlowJo, which is dragging the root file on first and then overlaying the query gate. If you right click on that overlay, there are options to show multigraph histogram traces. And so this histogram profile gives me a real easy way to phenotype the expression pattern of any region, right? Wherever you place this gate, you can drag this gate around in your graph window and it'll move and update the layout. So wherever you place this gate, you'll get the phenotypic expression pattern of that grouping of events. And then if you take your gated conditions and drop those downstream of the query gate, you'll notice that the expression of these or the distribution of this population of events is not 25% across all of the different conditions, but preferentially enriched in one condition or another. And this is how we can effectively explore this data space and get both the phenotypes as well as the distribution of events across your conditions without even using an algorithm to create a cluster. So combine this with the heat map where you turn on a particular color map, you pick a marker that you're interested in, and that will focus you on the areas of its expression, drag your query gate to that region, look at the layout editor for the expression pattern, and then your gating hierarchy for the distribution of events across your conditions, right? And I'm going through this really fast, but we're going to break down and compare these guys a little bit and give you some options for the clustering and dimensionality reduction we'll come back to in a moment. So if I cluster, right, and I get a whole bunch of populations, I want to be able to compare those populations. And before any other visual tools were available or any statistics, easy way to get these out, I could always enumerate the MFIs, the median fluorescence intensities of each cluster population, and that would enable me to call the phenotype. And what I've done over and over is to export the cluster populations that come out of a clustering algorithm as new FCS files, and then load them back into the workspace and put them into a group, because my table editor will batch across a group of samples very effectively to give me a statistical output. I then add the median fluorescence statistics and count statistic, plus all my experimental condition gates. That's the different conditions across which I'm questioning how are these populations distributed. And then drag all of those statistics into my table editor, and I can enumerate all the stats. And all I'm doing here is exporting to Excel and hiding the numbers and doing a heat map across each column to show the expression pattern across all my clusters for each marker. And then a heat map across each row here for the 4 different conditions to look at the distribution across the different time course and across the different compartments or tissues. So this is something you can do without any additional plugin. It's just using your table editor within FlowJo and then formatting using Excel's heat map so you can see the differences. Very effective way to share what are the clusters and how do they change. But more recently, as of about 10 months ago, we have this Cluster Explorer user interface, which allows you to visualize both the heat map and the clustering overlaid onto a dimensionally reduced data space, along with a profile of the clusters that's very simple to read, and you can gate within this profile. So I can actually gate for the cluster populations that are high or low and do combinatorial gating. So it has to express this and this, but not that, to isolate the populations that fit an expression profile. So this Cluster Explorer is something that you spin up after you have clusters from an algorithm that we'll go into, and a dimensionally reduced set of parameters. So in this case, my TSonique parameters. And then I'm picking in the middle section, what are the parameters that are measured that I want to heat map across and compare these clusters. So within this UI, you can actually click on the clusters in the list. You can click on the clusters within the dimensionally reduced data space, and that will limit what you see in the heat map and in the Cluster Explorer. What we call this is a principal component plot, right? On the y-axis is the relative expression of each marker, and then the lines connect each of the individual populations to show its expression pattern. So I'll go into this. I'll show you a bunch of individual images derived from this Cluster Explorer as we go through the different dimensionality reduction and clustering algorithms here. But this is a very effective way to explore the cluster profiles and what you get out of one of these algorithms within a dimensionally reduced data space visualization. So, but before we get there, let's take 2 steps back. to dimensionality reduction. I've said it before and I'll say it again, and anybody new to the field and high parameter data and these algorithms needs to know that dimensionality reduction just creates parameters. It doesn't actually create populations of events, but it does create two new parameters, embedding the events into a low dimensional data space. And the goal here is to give a low dimensional representation of this high dimensional data and preserve the overall structure of the data as much as possible. And different algorithms do a better or worse job of preserving the structure. We'll go into that. But bottom line is when you run a dimensionality reduction algorithm, the events with a similar expression pattern are going to group together into these different islands or nodes within this dimensionally reduced data space. And I'm showing this here on TSonique. This is 25 color panel that has all the major lineage markers in it. And I'm overlaying my manually gated subsets onto this data space. And what I want to point out is how well they group together, right? I mean, in this data set, the B cells, NK cells, monos, and CD4s and 8s, they all group together into these continuous islands, but you can see there are different peaks and valleys within each island that may represent distinct subsets within the lineage. And there are gray regions that my manual gating completely ignored, right? So manual gating is biased. You gate on a specific subset via a lineage marker expression profile. And wherever you cut that, everything that's not positive doesn't get into your system and isn't ever looked at. And so this is one approach to looking at everything, right? You're just looking at all the events, and each of these gray regions will also be clustered and represented in the total expression profile of all those populations at the end. Now, sometimes these islands will break apart, and it totally depends on the data and the seeding of the islands in the algorithm. And I'll show you an example of that. If they break apart and you see a non-continuous island that expresses CD8, well, there's probably something else that's different about those populations, not just their CD8 expression, but maybe some memory marker or something. So the problem with this dimensionality reduction algorithm approach has always been memory and computational power, because TSonique in its original form was very difficult to run on lots of events. It didn't scale very well, and the iterative nature as these events push and pull on each other and resolve out into these different islands is not conducive to parallel threading and high performance. So we were really limited by the number of events that we could push into the system when we did dimensionality reduction. And so if you don't have a very powerful computer, I do want you to think about, well, what do you dimensionally reduce for the experiment for the experimental question that you're asking? And I'm showing the same data here now, just dimensionally reduced and overlaid with my manual gates at different levels in my manual gating hierarchy. If I take all the cells in this whole blood experiment, I end up with the grands dominating the visual space that comes out of it. And if I'm not interested in those granulocytes, well, maybe I want to get rid of them, take away the CD15 positives, and now I'm left with the other lineages, and they spread out more effectively, and I can see now the different lobes or peaks and valleys within a lineage. And you can do that as deep as you want to limit the number of events and focus in and spread out on a population, spread it out so you can see the different subsets within it. And so it's up to you where you want to start looking. And you can do it at any gating level with downsampling and concatenation at that level in the gating hierarchy. If my question is only about CD8s and I want to use every CD8 event in my experiment, well, maybe I gate down to CD8s and just explore those independently of everything else. If my question is homeostatic relationships between lineages, well, maybe I need to do it at the level of CD45 positive immune cells. So just think about that, and remember, fewer events will run faster through TSonique and the other dimensionality reduction algorithms. And that is one of the reasons why we want to downsample and limit which populations go into the dimensionality reduction. Once you've decided on a level in the gating hierarchy that you want to dimensionally reduce, and you've created those parameters, this third parameter color map can really orient you with what's going on in your system. It'll be easy to find your major lineage markers and see the islands that correspond to them. And then you'll find features that, you know in two-dimensional space what they are, but you've never looked at them in multi-dimensional space, like this little peninsula that comes off the CD14 positive monocytes that has to be high for CD45RA and expresses CD16. Those are my CD16 patrollers. But you aren't used to looking at them. The more you, in this dimensionally reduced paradigm, the more you look at these TSonique continents and the islands that come out of your assays, the more familiar they will get, and you'll start to recognize features. And again, this will allow you to focus in on a particular area that expresses a marker of interest, and then you can circle that area and with a query gate, ask what does it express and how does it, how is it distributed across my experiment. And as an aside, for everybody that's using these color maps and wondering, to make those colors pop so much, just remember that in this implementation in FlowJo version 10, that the color map scale is dependent on the transform scale. So we're actually giving red to given for each parameter and blue for the lowest. And my connection's unstable, I'll just wait for a sec. But what we can do to change the color and make the reds pop and the green go to blue is use your T button, T for transform. Remove the high-end space here with the plus minus buttons or the max value. Adjust the width spaces to spread out the data. Usually I go to the left to spread the data out a little bit around 0 to make it closer to the bottom end of the scale, and then click apply for that parameter. And now I'm scaling the actual expression pattern of the color parameter that I'm interested in. And when I apply this scaling instead of this scaling, then the reds get redder and the greens get darker green, all the way to a blue for the low end of the scale. And I just want to bring that to you so that everybody is aware of the transform that can allow you to make those colors pop in the scale pattern.
Actually, quick question.
And then, yeah, please, Laura, go ahead.
Is it better to do that on the individual files before you concatenate, or should you do it on the concatenated file and then do all the transforms, or does it matter?
Honestly, I do it on the individual files first, because it's just out of habit. I transform everything as I go through and gate and look at my data files in the first place, because I know that by using this T button, I can get better resolution and see the inflection point between positive and negative better. And so I usually do that on the front end, and then at the back end, I'll go ahead and do it again to make something brighter. So it can be done on both ends. I do like to transform beforehand to some degree, and then afterwards, if I want to make that color pop, then I'll transform it again.
Okay.
It doesn't matter before or after, really. There are some reports about transform space affecting TSonique, but I haven't gotten into that. Ask me in a couple months, I will. But I don't have a lot of observations on that transforming before and trying different transforms yet. So once you have the data space, right, we can turn on the color map and we can query gate it, and we can also fire up our cluster explorer, and we can observe the, using these together, we can actually see how this gating is effective at limiting the clusters that express a certain combination of markers. And so here I gated on CD45RA high clusters. That would include B population in the bottom left, but then click the not button and exclude those CD19 positive populations that are down here, and they end up not being shown. And now I'm only looking at non-CD19, CD45RA high populations. Just another way that you can use Cluster Explorer, and I'm just showing this so you can see the gating limits the visual traces of each of those clusters and what is shown on the other visuals. You just click on any of these plots and copy them out as PNGs right into PowerPoint very effectively. Now, when we get to dimensionality reduction algorithms, I've been showing you all T-distributed stress burn plots, but there are a couple different options that we've added in a year and a half. UMAP is probably the most popular and is a very good alternative to TSonique, uniform manifold approximation and projection. TriMap is brand new. We've only had it for less than 2 months in the exchange, and EmbedSum also new. EmbedSum is actually laying out a FlowSum clustering output. So you have to run FlowSum first and then point to the FlowSum R file to create the embed sum. But TSonique, UMap, and TriMap are all valid options. TSonique does a good job of flattening everything and grouping events in a way that you can see them all. But it preserves local structure. That's the relationships between the different sub-lineage subsets within a lineage. And UMap does a better job of preserving global structure. That is the relationship between lineages. So when you look at TSonique, you cannot infer anything about the distance between or relative placement of the continent islands, but you can infer something about UMap and TriMap. It does a slightly better job of preserving the relationships between those different lineage islands. Now I'm going to go through each one, and we'll try to not take too much time because I'm already way over time. But this is UMap, and people have done this is TSonique. This is the algorithm components of TSonique, and people have done entire sessions at Cyto describing how TSonique was built. It's kind of a Frankenstein conglomerate of different algorithm pieces, and it really works quite well to dimensionally reduce and spread out your data. I'm not going to go through this. You can actually read a really nice blog from the towards datascience.com by Nikolai Oskolov on exactly how UMap and TSonique work and what's the differences between them. But when you start reading about this, you will get some basic concepts like perplexity controls the number of nearest neighbors in the algorithm, and the gradient descent is controlled and the comparison is controlled by this Kullback-Leibler divergence. And I don't want you to focus too much on this, right? Because I don't understand this math. I don't expect anybody that's an immunologist or a bench scientist to really understand the details here. But we can start talking about, well, how has this algorithm progressed? There are a bunch of challenges to TSonique that have been overcome over time. The first one I've already mentioned, TSonique is non-deterministic. So it depends on a random seed number. At least the Barnes-Hutt original implementation was a random seed, which means you got a different plot every time, even on the same data. And so what we've done in FlowJo v10 with the current TSonique implementation is not a plugin. It's actually a standalone platform right next to the plugins menu, is set the seed number to 3. So that means you run TSonique with the same settings in FlowJo v10 on the same data, even on a different computer, right? As long as the settings are the same, you'll get the same plot, which is as deterministic as we can make it. Now, this does not mean that you can run it on different samples, right? Running on different samples has different data, which means you get different plots. So you still need to concatenate all those samples together first before you run the dimensionality reduction if you want to compare multiple samples within the same common dimensionally reduced data space. The second challenge is that these TSonique embeddings, this algorithm is so computationally expensive, it really had to downsample before running TSonique. And even 2 years ago, 3 years ago, with the first TSonique implementation, it was a single core Barnes-Hutt TSonique. And I couldn't scale it up of about 1 million or 2 million events on my machine. It would just take too long to run. And so to speed up the TSonique calculation, there was a paper that came out describing this fast Fourier transform accelerated interpolation-based TSonique called FitSonique, which was faster, but also led to more memory consumption. As part of that paper, they developed a new or implemented a newer, faster way of looking at nearest neighbors called an Annoy library. And that is actually options that we have incorporated into our TSonique platform. So you can turn on the faster FitSonique gradient descent option, as well as the faster KNN Annoy library for finding nearest neighbors, definitely speeding up the process. Then there was this problem with too many repulsive forces with large data sets, which means you just got a ball if you tried to TSonique over a couple million events. And Anna Belakina, along with our bioinformatics team at FlowJo, have collaborated and come out with an OptSonique, a modified algorithm that does a couple different things. It actually looks at the algorithm and makes some choices during its running, when to stop different sections of the algorithm and initiate the next step. And what this does essentially is give us the ability to resolve these multi-million data point TSonique embeddings. So map with tens of millions of events. And then finally, I already mentioned it, but TSonique doesn't really preserve the global geometry of the data. Relative placement of islands for lineages don't mean anything. But UMap and TriMap do a better job of that. And so if you want to observe the global geometry. You can run TSonique to flatten things out first, then you can run UMAP on the same data and get two different views of it, right? The dimensionally reduced data space, these UMAP and TSonique derived parameters are just giving you a visualization that you can then use downstream to gate or to overlay cluster populations. And so the paper is a Nature Communications paper, the Abbvie paper. It is one of the most downloaded papers in the first quarter this year. So people are very interested in using this tool. All the details can be found there. Bottom line is that once we start using Abbvie, we can resolve, in this case, 20 million events into these nice discrete islands in the TSonique plot. And then we can scale that up on a high-powered workstation and run 40 million events. And this is really important if you want to merge many samples together and look for rare populations that could be lost during the downsampling approach. To be honest, 40 million event took 7 hours to run, so it is not something to be undertaken by the faint of heart. But I do have individuals trying to run billions of events. How long that's going to take, I'm not sure. And if we actually turned on some of the options that are available in the TSonique graphical user interface, in particular, changing the k-nearest neighbor algorithm and the gradient algorithm from Barnes-Hut to TSonique, I'll go through this in just a second, a little bit more. We can speed that 27-hour processing up and get it to drop down to about 13 hours. So when you spin up TSonique, you just point to your concatenated file and you'll be prompted to pick the parameters you want to use to dimensionally reduce. All my compensator parameters here. The Abbvie learning configuration is automatically engaged here. We're using Abbvie as the default now, and you can link to the paper right here within the graphical user interface. Actually, if you change the gradient algorithm from the drop-down menu to be Fitsony, there's a link to that paper as well, so you can get those references. And then you click run to just run this, right? Don't worry too much about these other options. I have yet to see with the defaults an image that does not open up into this continent-like structure. You can change the number of iterations, as the number of cycles the algorithm goes through. And if you don't give it enough cycles, then you might end up with a ball or a line. Things don't open up into this nice map until later on in the calculation. The perplexity is a dial controlling the number of nearest neighbors, so how many events each event is pushing and pulling against during each cycle of the calculation. And the learning rate, this is essentially how fast you step towards the solution. If you make it too high, you can get into a bad valley of a solution and end up in the wrong place. So you don't want to make that number too high. But bottom line is that the learning configuration, when you set it to Abbvie, it automatically chooses the rate and then changes that over time as part of the Abbvie calculation. So I don't think you can go very wrong just using the defaults and clicking run. That's what I've done here. And this OMIP 060, it's a 30 color panel that I pulled off a Flow repository to play with. And it spreads out quite nicely. And I think I'm showing phenograph clusters on top overlaid there. Now, UMAP is another option. And I've mentioned a few of the differences, right? It does a very different mathematical process, but still has the same iterative nature to solving the low dimensional embedding. That is the UMAP space that is created here and where the events end up positioned. So UMAP uses a neighbor style graph. It uses these simplices, this complex of simplices, which are geometrical shapes, and then to produce this fuzzy topological map, links all the simplices together to create this global topological structure. And then it tries to solve and relate the topological structure of the low dimensional embedding to the topological structure of the high dimensional data. And you know, I'm not going to go into the math. I do have a slide on it, but it's not my you know, expertise. So how does it work for me downstream? Well, it definitely gives meaningful representations of the data. So here is the same OMIP run through the defaults of my UMAP program or algorithm. It does a better job of seeing the multi-branched continuous trajectories of the relationship between cells. So how these islands are connected with each other. It's faster than the Barnes-Hut TSonique and more comparable to the Fitsony implementation, and it scales better with higher number of parameters. So UMAP's being used in the multi-omics and single cell RNA space quite a bit. because you can actually input hundreds or thousands of parameters into this algorithm, which TSonique you would need to do a pre-dimensionality reduction like principal component analysis and then feed those in. You can't do it with hundreds of parameters. It's just too computationally expensive. The output is preserves more global structure so that the islands that are similar to each other group together and aren't found in different areas all blocked apart. I'll show you an example of what that means in a moment. And you can now produce multiple components. So I'm not limited to just an X and Y. I can make 10 or 20 different components similar to principal component analysis, and then use those multiple UMAP components downstream for whatever I want. So you are prompted to select the same type of parameters. And for any clustering algorithm or dimensionality reduction algorithm, you have to select the parameters that you want to use. You don't have to use just Euclidean distances. You can actually use several different distance metrics, and you can control the number of nearest neighbors. We've tried to set the defaults to be what most people will need for flow cytometry data. And so leaving the defaults and clicking OK is how I get this map, right? If you want to start varying the different number of nearest neighbors or taking a look at the distance metrics, you may or may not be pleased with what you get. I've done that a little bit. Just as an example here, the Euclidean is the default, which gives me this map. But if I switch to Manhattan or cosine, I get slightly different maps. They're kind of flipped along the y-axis. But immediately I know that I don't like cosine because just the way things are distributed. Maybe I like Manhattan, so I can take a look at that one. Vary the nearest neighbors, run UMAP a couple times. I see that the lower number of neighbors gives me wider distributions, and the continent islands seem to be closer together. This is the default, looks pretty nice. And then the clusters are even tighter with the more nearest neighbors, right? So keep 15 nearest neighbors and vary the minimum distance. The default is 0.5. It's a nice map. Wow, what a difference makes when I drop that minimum distance, right? Tightens up these islands quite a bit, gives me a lot more space between them. And the opposite occurs a little bit, not as dramatically, with the 0.75 distance. And so what this means is I could use a low minimum distance to get very effective separation, right? If you look at the blue and pink clusters here, they're grouped together in the middle, and I couldn't individually gate that region apart very effectively, especially over one that's all mashed together, but I can do so right here. And there's no reason why I can't run dimensionality reduction, gate on an island, and then run a different dimensionality reduction algorithm to spread those events out again. So I could run UMAP first to subset lineages very effectively, and then run TSonique on that subset to spread it out and use the entire data space downstream. Now, whatever algorithm you use, right, TSonique or UMAP, I just want to point out that they are still iterative processes that take a while to occur. UMAP is a little bit faster as you scale up, and a lot faster as you scale up in the number of parameters, but they're still doing the same thing. They take a high dimensional data set, and in the case of TSonique, present this distribution. It converts it into a scaled distribution. And then you take the solution, that is the embedded data set with some events here, and convert that into a distribution and compare the two distributions and say, how similar are they? And this relative entropy comparison is controlled by that Kullback-Leibler divergence. And it's an iterative process. This is the iteration. This is the solution we're trying to match right here, the original data turned into a probability distribution. And we're iterating by moving the events, converting, and then asking, is it close? And then moving the events again, converting, and asking, is it close, right? And we're doing this 1,000 times. Or with obstinate until some specific metric of separation hits a certain threshold, and then we stop the iterations because it's as good as it's going to get. UMAP does the same thing. It just uses these fuzzy simplical complexes instead of distributions, instead of these probability distributions, and it uses a method called cross entropy to compare the two fuzzy distributions. But it still is iterating the solution over here and trying to match the simplex complex from the low dimensional data space to the simplex complex from the high dimensional data space. I hope that helps make you think a little bit more about, these algorithms, very different in terms of some of the math that they use, but the overall approach to embedding events in a low dimensional space is very similar.
Hey, Tim.
In their iterative process. Yes, sir.
I just wanted to let everybody know online, and I know we're going long.
Yeah, I'm sorry.
Which is fine. I mean, as long as Tim, you're okay hanging on.
I'm fine. I feel bad for everybody because I'm.
Well,
there we go.
the beauty of this is that this is being recorded. So if you need to leave, great. That's totally fine. Just come back to either this YouTube link, or check back on the ChUGcytometry.com webpage, and you'll be able to review the link, review the video then again. So we're going to keep chugging along here. We have our list of questions that we're going to hit at the end, but I think it's a lot of information. So we gave Tim an impossible task to try and wrap up within the hour, but I think it's important enough to continue on here. So we're just going to let things run a little bit longer. We'll get through the rest of Tim's slides, and then start with the Q&A session.
I will try to speed things up. I know I'm
that's okay. No,
on some of these, but
it's great stuff. Great stuff.
Thank you. So I'll just keep going, and thank you guys for your time and attention. Absolutely wonderful to have you all with us. TriMap, newest version. It is a modification. Well, it preserves global structure quite well, and I don't like what it actually came out of OMIP 060 here, but bottom line, it uses some PCA embeddings to kind of preserve that local structure, and initializes on that and uses triplet information from the high dimensional data set to refine the graph over time. It does give you the same kind of meaningful representations as UMAP and TSonique, and it is an alternative that is now available as a plugin. The only downside here is that it does require the R environment and libraries within R to run, while UMAP and TSonique are both standalone. They do not require any additional setup or R. If we just look side by side at these three different representations we get of the same data set using the defaults of the algorithm, Opsony, UMAP versus TriMap, you can see the differences very clearly, that these global structures are more preserved, that the areas that are closer together when phenotype actually group together, and that is not true for Opsony. And I'll show you on the next slide that these different clusters are actually distributed in different areas of the TSonique algorithm, or the TSonique data space. And so you can't guarantee in TSonique that things that are close together, these different islands, are actually related to each other. Just within an island, you know that those samples are very closely related, but you can make that assumption with UMAP, that if it's closer together, they're actually more closely related. And if we now fire up Cluster Explorer and we gate just on our CD8 populations, you can see that quite well in the different visualizations, where the CD8 profile has got 7 different populations based on flow sum. And we now have these CD8s that are distributed in different areas. And I often tell people to look at your TSonique and then imagine a Z-axis where these touch in the back, along the Z-axis. So you can connect them in another dimension, but we've just flattened out the data here. So which one is best for you? It's really up to you how you want to present this stuff. If you want to gate on specific clusters, TSonique is probably going to give you a better, flatter representation in two-dimensional space that you can actually circle these islands with. But UMAP and TriMap are going to group those events that are more similar together and give you that additional global structure information. The last option here is embed sum. I won't go into it, but it is taking a flow sum clustering output and actually projecting it into this two-dimensional space, or embedding it into two-dimensional space. And it's actually using the grid map output of the flow sum algorithm to position the clusters. And so it ends up with this like square looking profile with all of the clusters kind of overlapping onto each other. But it is another option. It's a very new algorithm that we have just put in as a plugin. So you've got this clustering, or this dimensionally reduced data space, and I've been showing you clusters just visualized through the Cluster Explorer in all these images. But we create clusters for a purpose, and it's to have some subsetting of populations downstream of our events. And to explore those populations, I want to make just a couple points here. If you run a clustering algorithm, it does a really good job of capturing transitional intermediate states. And if you're hardline gating through these distributions, like CD94 here, if I just cut with a positive gate somewhere where I would normally and define everything as positive to the right, everything to negative to the left, I'm not doing that complexity justice. I'm cutting through the low expressing populations and chopping them up and defining everything in the same bin, whether it's mid, low, or high. And so I really think that the clustering algorithm is a great way to explore high dimensional data if you don't know what you're looking for. And if you're afraid that your hardline gates are actually chopping up interesting populations, you will get a whole bunch of populations downstream. You can visualize their expression profiles and you can determine how they are changing across your conditions. And so why are we going to cluster? For just those reasons. You aren't limited to two-dimensional plots, and you are no longer applying your own investigator bias when you're creating and drawing gates. We often apply bias in the same way over and over and over again, however we're taught by our instructor on how to gate. And so, hardline gates, they cut through the continuously expressed antigens and you only gate for what you know, which means you can miss novel populations. But a clustering algorithm is by far and away more objective. It's guided by the same assumptions for each given algorithm, and it classifies the events into populations defined by all dimensions simultaneously, not one at a time with hardline cutting through for positive and negative. And so again, it captures transitional intermediate expression states more effectively. It has the potential to identify populations that will be ignored or missed with your manual gating. And it could give you more information in detail regarding the complexity within a known population. You can imagine you gate for a specific subset and then you manually, and then you run clustering and you get a of that. Within that subset, there are 3 different populations. Well, what are they? How do they differ in their expression pattern of given markers? You can figure that out. And you may be able to find that your manually gated subset is not just one, it's actually multiple populations using a clustering algorithm downstream. There are 3 different main clustering options that I will promote. XShift, which uses fast k-nearest neighbor density estimation. Phenograph, which uses a nearest neighbor graph and partitions that graph into communities, which are the cluster pops. And then self-organizing map, FlowSum, which creates meta clusters from that map. And you have to actually specify how many meta clusters to get out. So same data space, TSonique, dimensionally reduced, overlaid with the cluster populations and visualized in my cluster explorer. My point here is only to show that they cut differently, right? Certain algorithms will group all events the same. Others will actually subset them. You can see the difference here in the middle. It's because 1, 2, 3, 4, 5 populations within this elbow, but FlowSum only captured 3 and XShift only captured 3. So it's really hard to say which one is better with a direct comparison that's not more granular. But I want to break it down and just say XShift is probably my favorite right now because it uses density gradients to define the populations. It's developed by Nikolai Samusik and Gary Nolan's lab, and now Nikolai works for us at BD, and he has written this as a standalone plugin that does not require R. And you can vary the number of clusters by changing the K, the resolution of nearest neighbors, smaller, and you get more clusters and more closely chopped up populations, but it's more sensitive to stochastic variations and batch effects and such. But the beauty of this algorithm is not only that it's a standalone algorithm, doesn't require you to set up or install anything with respect to R, but it also automatically estimates the optimal K as finding the switch point mathematically, and uses that K as the input. It depends on the number of parameters that you select. And so using XShift, we can run this clustering algorithm and it spits out a whole bunch of populations, and we could overlay those populations and spin up Cluster Explorer and obviously explore those clusters. And I really like XShift, and I think that Nikolai is really onto something with this KNNDE approach. The benefits here are that it's fast. It's a very fast program, and it does not require you to put the number of clusters you're going to get out. It's going to find that. It is looking for small rare populations by default, and it doesn't require R, but it doesn't give you any visuals or statistical output from its run. Those will be visualized in Cluster Explorer or in your layout editor and your table editor and such. Phenograph, I like this paper because it actually created a new algorithm and applied it to a real disease state model and asked a question. It's a cell paper that you can pull down. You do the same things, point to the parameters that you want to use, set the K, but you don't need to actually change that because it does, similar to XShift, estimate that number of communities and find the number of clusters as part of the algorithm. And so you don't actually have to say, how many clusters do I get out of Phenograph? It'll find what it finds, and you can use that as a starting point for the next algorithm, which is FlowSum. FlowSum, oh, it's okay. Phenograph, it's a little slower than either FlowSum or XShift. And it doesn't require you to specify how many populations come out of it, but it does require the R environment to run. So we are leveraging R on this algorithm. FlowSum was the first really nice clustering algorithm that we got maybe 2 years ago, little more, into FlowJo as a plugin. And FlowSum's nice because it actually uses a self-organizing map, produces meta clusters. You have to actually specify the number of clusters here. And number 39 is how many populations you're going to get out of it. But the problem then was always like, how many clusters do I create if I have to know what the complexity is? And so what I've been doing for a while is running Phenograph or XShift first, getting a cluster number, and then inputting that as the number of meta clusters into FlowSum. And FlowSum, people really liked it because it actually, it's R-based, it requires R, right? But it's super fast and reproducible. And it also comes out with the visuals and the statistics table that you can access as derivatives from the R environment. And so this is a picture of some of the visualizations that come out of the FlowSum algorithm. In addition to the cluster populations, you get this minimal spanning tree. You can actually turn this into a grid that EmbedSum is based off of, and these radial star plots that can tell you, well, what's the expression pattern of the clusters. And then this is obviously a relationship of expression pattern and a dendrogram showing the relationship of the clusters there, which is kind of nice. But which one do you use? Again, I'm not totally sold on FlowSum being the best because it doesn't actually try and find the number of clusters. And so I already need to figure that out first. XShift I really like because of its density estimation, and Phenograph, well, I really like the way it cuts up things. So your guess is as good as mine still on which one is best. Try them all and explore for yourself. Or if that's not your purpose, then just use one or the other and go with the flow downstream and use those clusters.
Have you used Phenograph on larger data sets? Like
it chokes up above 3 million on a fairly decent PC workstation with decent RAM. So there is that limitation. It's a good point to make. That's why I call it medium fast, because it takes a lot longer than FlowSum to come back with the clusters. I haven't pushed it up beyond where my computer limits me in terms of memory. If you have more memory, you can push more events through it. But it's also using R, and R is a single threaded calculation. So it's only using one core. So there's only many things you can do within that environment to speed things up. I would only try it on 1 or 2 million events, realistically. Otherwise, you're probably going to run out of memory. FlowSum, I think you can scale a lot further.
In my experience, I was running out of memory with like 2 million events.
Right. And you're probably on a 16 gigabyte 4-core machine.
Yeah, I don't remember what I had.
Laptop or something like that. If you had double the memory.
Well, it was on a desktop. But
yeah, well, it just depends on the specs of the machine. If you're really going to scale these up, and especially you may need to get a high performance workstation or go over to the bioinformatics department and use a little cluster. Cluster on the cluster would be the recommendation. But that is a good point. There's always memory limitations. And if you run out of memory, well, scale it down in the number of events and or number of parameters you're considering. So now we've got this, I promise we're almost there, guys. We're almost there. Thanks for sticking with me. Now we have this population you find and you're interested in it. What are you going to do with it? Well, you don't have a manual gating hierarchy yet to identify that population. And so the question then becomes, how do you sort this population or populations you've identified as interesting using a clustering algorithm and dimensionality reduction exploration approach, right? All you have is a phenotype. and a population of events. Now, we've actually come up with a solution for this, and I won't take credit for it. I'll give Nikolai Samusik full credit for merging all the math between two different approaches to generate gating, a gating strategy to sort or purify a population of interest. So there were two publications that came out, GateFinder and HyperGate, and they did slightly different thing. And what Nikolai did is merge the math between those two. And actually he's the author on one of these, and allow us to create a HyperFinder plugin. So HyperFinder will actually take half or 70% of the events in a gated cluster population and train a gating hierarchy to purify that population, and then validate that hierarchy with the other 30%. You can change how many go into the training and validation data sets. And you can create either series of convex polygons or hyperrectangles that are optimized for the simple, most direct way to purify that population. And HyperFinder does those double checks of how clean is my population, recall, and with a harmonic meaning of which, well, okay, precision recall, and you can generate an F1 score, which is like, if you gated and I gated, how many events were overlapping in both our gates? How much do they match? So an F1 of 1 is perfect, and F1 of 0.75 is 75% overlap. And then it actually does this iteratively and removes redundant gates and finds a gating hierarchy to purify that population. And so you end up in FlowJo with a series of gates that are actually from the HyperFinder plugin. And this gating hierarchy, some of the gates you will see you would never draw as a human, actually purifies, in this case, down to 0.95 F measure, which is 95% pure, this sub 1% population that I was pointing to in 8 gates deep. And so you can actually say how many gates deep do you want to use, depending on your sorter. Set the F measure, a higher number versus lower number will give you better precision versus recovery. And then you can set whether you want to make convex polygons or hyperrectangles, and click OK. And it'll go through this iterative process and generate these gates for you. And you're like, okay, well, sure, now I've got gates in FlowJo, but what is that going to do with me? If you aren't aware, we now have, with a right click on that sample, the ability to export those gates now back to FACS-DIVA. And so you can actually export your HyperFinder derived gates to your DIVA experiment, exported from DIVA, and it'll add this gating hierarchy as plots in a new worksheet that you can then take that experiment back to your sorter and actually purify this population. So we have this workflow now that enables computational derived sorting. Find a cluster, right, using your dimensionality reduction and clustering tools. Use HyperFinder to, dimensionality reduction and clustering tools and visualization tools. Use HyperFinder to create on that cluster to create a gating strategy, and then you can go back and actually overlay that gating strategy onto your original samples to validate and use it downstream to sort. And so, what does it look like in a workflow? Talked about to create populations, right? And then we can find that population, visualize it in a dimensionally reduced data space, see its expression profile. If you double click on that gated population, that is the cluster, then you'll clean up the cluster by gating away any of the events that are outside of it, and then run HyperFinder. And HyperFinder will generate the sorting gates. Then export those to DIVA, go back to your sorter, and you can actually sort those events. You can also use HyperFinder to just shorten the gating strategy of your already traditional sorting gating hierarchies, because a lot of people use bias, and you might have 8 gates deep to purify a population. You run HyperFinder and it finds that population in 2 gates because you weren't considering the right parameters to sort it. And so there are 2 use cases here. One is discovery to purify a population that you found from a clustering algorithm and high dimensional data. And the other is to actually try and purify this population better than your manual gating with bias originally, that you had set up originally. So when you have this sorted population, of course you want to look at it functionally, you want to look at it downstream, and you could take these populations on the Symphony S6, you can sort 6 populations at a time and drop these onto a gene chip. This is the Rhapsody system that BD offers, and our SeqGeek application to look at single cell multi-omics data. Once you have those sorted populations, you can drop them onto a chip, the cells sink into the well, you pack with a bead, you lyse the cell and all the RNA gets captured. You can stain those cells with oligo-tagged antibodies. I'm sure you all know this as the Abbvie approach, antibody tag or sequence tagged antibodies. And then you capture those sequences as well. And this enables you to look at both protein and RNA in the same single cells and get hundreds or even thousands of measurements that you can then work with the same or similar dimensionality reduction approaches and do differential expression analysis to get gene sets. There's a nice little paper by Kobach et al. On how to use TSonique within higher dimensionality data for single cell multiomics and transcriptomics. And I'm showing here that I initialize with principal component analysis to generate multiple principal components and then use those principal components as the input for TSonique to retain the global structure as well as separating out the local structure, which is a nice trick. I learned it at Cyto from a talk from the Broad Institute, but if you're new to multi-omics or single cell transcriptomics, it's worth a read to get some of those principles down of how we might approach the data analysis. And in many systems, the raw data to usable interactive file can be a challenge here. And they've figured all that out, and you have a pipeline that's offered that you just upload your raw data and it spits out a molecules per cell normalized expression matrix that you can then explore interactively within FlowJo or SeqGeek, that is. So that's all the slides. In summary, these plugins that we use are facilitating interaction with bioinformatics programming environments like R and enable us to use algorithmic tools as bench scientists that even if you have limited bioinformatics experience, right? It's a point and click interface to utilize these algorithms. Using plugin tools in that proper workflow that I've described, which is cleanup, concatenate, dimensionally reduce, and or cluster, and then gate apart and compare, allows you to potentially find novel populations and discover things that are complex in a new high parameter system that you may not be familiar with, right? If you don't know what you're gating for, you can't gate for it. If you aren't familiar with that population, you won't be able to isolate it manually, but you will find it with a clustering algorithm. And then we've integrated FlowJo and BD Diva to some extent, enabling the transfer of gating information between the two platforms. So FlowJo can export gates to Diva. You can also export gates from Diva to FlowJo or read in gates from a Diva experiment. And this is really going to enable you guys to construct gating hierarchies using Hyperfinder to isolate a high parameter population of interest in FlowJo, and then push those gates out to your Diva experiment so you can sort and purify that cluster. And then finally, if you aren't familiar with it, the BD Rhapsody with both protein and single cell RNA measurements is an amazing system that is going to revolutionize the industry. It provides targeted panels as well as full whole transcriptome experiments. You can customize the panels, and you can, with any antibody in the BD catalog, tag it with a sequence and include that antibody sequencing as a measurement, as well as the RNA sequencing. So you get true multi-omics analysis. All of this work is really done by the smart people in my group. So my bioinformatics team is fantastic. Richard Halpert and Sahid and Yosef are my go-to guys with questions. And if you do have questions that I can't answer today, I'll be on the line with them and get them answered for you by the end of the week. My advanced technology group, Nikolai Samusik, has been amazing in producing algorithms and plugins for us to sort these computationally derived populations. And Joe Trotter is the main driving force behind the Cluster Explorer UI. The algorithm and data authors here, there are many of them. I'm sorry, there's countless others that I should give thanks to, but these are the people that have written those algorithms, and then the data that I was using from the OMIP 060s from Mario Roeder's lab in atomic Thomas Lichty here. So with that, I'll say thank you. I know we should have given this an extra half hour. I'm still within the hour and a half, but I'll open it up to Ryan and Laura for questions and see what I can answer for you guys to clarify any of the points that I've made here. If you have questions for me specifically, my email again, top bottom left corner, timothy.crawford@bd.com. And thank you all for your time and attention today. Ryan, do you have questions for me?
Yes, we do. Yeah,
Awesome.
Thanks,
Yeah.
Tim. That was great. Yeah, so I think there was kind of a flurry of activity while you were discussing the whole downsampling and concatenation part of it. And I think it's, fairly typical questions that we get, but I'll read a couple of them and try and maybe weave them together, and then you can tackle that as a group, perhaps. So a lot of it went along the lines of having different numbers of events in different samples or in different groups. So if you have a couple of treatment groups and whatnot, how important is it to have a normalized set of events per sample? And then B, I would add, what if you have the ability to kind of downsample a bunch of samples within a group to a reasonable number of events, but there's, that one outlier where you had, 1,000 or 2,000 fewer events. You still want to include it, but, it doesn't really fit in nicely with the automated approaches. So do you have to kind of add that in as a one-off or what? So
you have to make an executive decision. And I think the statisticians will tell you if you have too few events, you're just looking at noise. So with bad sample quality issues that just have few events in the gated subpopulation you're interested in exploring, you may want to drop those out of the analysis because it's just adding additional noise. It's not mandatory to balance anything. And if you know what you're putting in, even if it's 67.2% sick versus 32.8% healthy, as long as you know what you're putting into the system, you can still get that answer downstream, which is how do those populations change across your experimental conditions. But it seems logical to me to balance everything. And so you can do sequential downsampling and sequential concatenation, where you could concatenate one study arm together using your 60 healthy patients, and then the other study arm using 100 healthy patients, taking the same number of events from each, and then downsample the larger to the smaller, right, to balance them out as 50-50. You're still looking at different numbers of patients, each one is represented, but you're still balancing the major question, which is how do the sick people differ from the healthy people? So I don't think there's any rule that you can make, that I can make, to say that this is the way it should be done. And if you want to use every event, even the crappy samples, and just push them all together and the numbers are what they are and you explore it, no, I'm not going to complain until I have to review your paper and ask, well, why didn't you normalize these things? But you see a lot of that in the literature and in presentations right now. So I like to promote downsampling because it makes sense to me to have equivalent numbers. That way, there's not one or two outlier patients that are really driving the distribution and the clustering. And it's really easy for me to interpret with the numbers downstream. But it's up to you how you want to approach that. The boss will say, well, keep as many samples as you can so we have the N, right? That's high enough. So we can write in, we have this many patients and this many patients of these different cohorts. But the statistician might say, oh, drop those crappy samples and just pick the 8 from each group that have the best numbers and explore with that. And Mario Roeder told me a long time ago with the original TSonique, he's like, this isn't really an analysis approach. This is a discovery approach. This is something you do not necessarily like gating and just pulling out the numbers, but you're exploring a data space, trying to find novel populations, and then you can follow up with those using manual gates. And I think that's still true, even though we can now look at more events. It is a discovery approach as much as it is an analysis approach. So don't expect so much from it. TSonique is just a visualization, right? It doesn't even do much for you in terms of creating populations. So don't expect too much from it. I hope that makes sense. Balance to me makes a lot of sense because I can just grab those specific numbers. 25% across the board is a lot easier for me to interpret, right? If I have 25% across the board, then having this one be 20 and 30 and 40 and 15. But you do what you got to do to use as much data as you have and preserve all of those events.
And I guess
Roundabout answer the question.
Yeah, no, that's good. No, in terms of like downsampling to a certain degree or number of events, really the only reason to downsample aggressively to a certain number of events is the computational costs. Otherwise, you'd want to keep as many events as possible. Is that true
Definitely.
or no?
That is very true. Yeah, there's no reason why I wouldn't want to keep every event as long as I can balance the system and still compute the outputs in those algorithmic processes. And yeah, I've been showing you all the stuff I'm showing you is 300,000 events, and it takes 5 to 10 minutes per calculation for TSonique or UMap to produce any of those maps. And so just to give you an idea, on my 4-core MacBook Pro with 16 gigabytes of RAM, each time I dimensionally reduce with 300,000 events, it takes me 5 minutes. That's not bad, but when you scale it up to 2 million or 3 million events, you might be waiting hours. And if you scale it up to 10 million events, you run out of memory and it just crashes and doesn't give you an output. So you have to work within the limitations of the compute power that you have, and then within the limitations of the data. If I want to include every event and my worst sample has 5,800, well, I can downsample to 5,800 to balance it, or I can make that executive choice where I'm going to take fewer events and just try to balance the sick versus healthy arms of the study in some way. But I can't give you I haven't seen anybody say that this is the way it is done and this is the way you should definitely approach it. I'm giving you my opinion here on what seems like a logical approach to me.
And just to reiterate a point that you made earlier, since there's so many good points that you made, but since we're on the topic of downsampling, you did mention that for newer users who are just starting out with this and trying to learn and play around with these things, it is a good idea to start with a very small number of cells so that you're not waiting forever to test out all of these different things. And then once you're more comfortable with the process, then you can go ahead with a much larger sample set.
And I'll give you an example of that. You know, my wife works at the bench and she's got some high parameter data. We've been using XShift and UMap to explore. And the first time she put all the data together, she did her classifications, but didn't make an ordering keyword, right? And then when she downstream, she was like, oh, what was the original order of my files? I can now group them, but I can't put them into the original order. So when I look at sample ID, I can't see it. So why not add in another column and give my order so I can always get back to this original order the way I want to look at it. And until you know what you need to look at downstream, don't scale it up to millions of events and wait for an hour for dimensionality reduction. Do it on 50,000 events. Dimensionally reduce real quick. Make sure you can pull apart the way you want to pull apart, and then scale it up and concatenate again with all the events. So yes, I agree, Laura, that's really important to make sure you know what you're doing with the concat step before scaling up and waiting for too long on the dimensionality reduction steps.
And is it something you said led me to believe that perhaps any of the algorithms that rely on R would run single-threaded?
Yes.
Is that true?
As far as I know, I'm under the impression that it's a single thread. And it's not even, actually not necessarily everyone. I think, I'm not the R expert, but whether it's R or not, the problem with dimensionality reduction is you can't parallel thread certain iterative parts of the calculation. So when we go to this iterative process, you can't start the next thread, you can't start the next calculation without actually having the solution for the first one, right? So we create a low dimensional embedding convert it to a probability distribution, ask, is it similar to the high dimensional probability distribution, and then modify the probability and do it again. So you need the solution for the comparison before you can run it again, which makes parallel threading in either one not as effective, right? So you can only parallel thread to a certain degree, and just speeding up the number of more cores will make it run faster, but only to a certain degree, if that makes sense.
Okay. Yeah. Somebody asked specifically about phenograph not running multi-threaded in FlowJo.
Yeah, well, it's not, because it's running in R, and I'm quite sure that that is a single thread.
Okay.
And there are in R. If you have trouble on a PC running phenograph, email me or email my tech support team. We have a document sometimes the path to saving the files with the RTools package isn't correct for phenograph, and it fails specifically on a PC because of the RTools setup. And we can fix that for you and get it to run again. But that's, when you introduce R as a whole nother computational environment, then you have a bunch of dependencies that I can't control on the FlowJo side of things. And so it becomes a little more complicated. TSonique and UMap come with everything you need. TSonique is actually native. UMap download as a plugin, but it does not use R. It's actually running independently of R. Phenograph is definitely R dependent, and there are kind of limitations to that, and parallel threading, I believe, is one of them. So it's not that fast compared to FlowSum or XShift.
Great. Somebody else asked a question here. Not exactly quite sure what it means, but just I guess using TSonique, TriMap, UMap, and doing that based off of uncompensated data or compensated parameters.
I'm always doing it on compensated data. In theory, you would still be able to separate, but I'm not going to promote that approach.
Okay.
I would prefer that you use the compensated numbers. I have never done that. I should give it a try sometime and see what the effects are. And you can.
Yeah. And then actually somebody had a question about the Rhapsody. In terms of cell recovery, I guess compared to the number of cells that you input into the Rhapsody versus the number of cells that you're actually outputting as single cell RNA-seq data points.
How much do you lose?
Yeah.
Recovery is pretty high on this, because you can load 20 to 40,000 events, and you can multiplex 12 samples. And I don't know what the actual metrics for recovery are. Can't say that I've seen those numbers, but it's fairly high. So you get thousands of events back out with lower multiplet or doublet rates than you would see on our competitors' box.
Okay.
And the recovery is very high because you just wash a buffer. This is a magnetic, the capture all has to do with magnets, and you wash the cells through, they fall into the wells, and then you wash away the rest of the buffer, load your capture beads, they get magnetically pulled in, and then when you lyse, all the RNA and stuff gets captured by the bead. And so the actual recovery is then you remove the magnet, wash, resuspend, you actually bring the magnet to the top, the beads come up, and then you wash everything out. So virtually every bead that goes in comes out of the system through a little port when you wash them out. And so there's very little loss there in terms of what you put into the system and what you get out of the system. And I think the bioinformatics has been worked out so that when you get that raw FASTQ set of files, that you can very easily input that into a pipeline and get out your expression matrix, which I think is one of the biggest benefits over some of the other approaches which have to merge multiple sets of data reagents from different locations together and then have a custom bioinformatic pipeline built to call the sequences and actually get your single cell expression matrix. The other point I'll make here for anyone that's interested in this technology is that the cell capture beads that bind all of your RNA and sequence tag protein antibody information, These are hard and can be, after you generate the cDNA, these can be stored, so you can subset your experiment, run an experiment, and then go back in and probe that same set of beads a second time or a third time with a new panel of markers. And that's really important because some of the other options are gel-based beads that disintegrate, and so you basically have one shot. There's also a whole bunch of QC steps that are involved in when you load these cells. The Rhapsody is a big scanner that does cell counts and visualizes whether you have cells and beads in each of these wells. So you take pictures of this cartridge as you load the cells and you load the beads, and you get a viability count, you get a doublet count, you get metrics at each step, so that when you go in to the sequencing step with your beads and your library derived from those beads, the sequencing that you're doing, you know that the quality of the reads is already there and the quality of the cells was already there. The problem with some of the current systems is that you get what you get out of it when you get the bead and the cell together, and then if it's bad data downstream, you don't know exactly what step things went wrong. And so they really have a better quality control system and a better bioinformatics system, enabling users with less experience in the genomics field to get started with and process these cells through the multi-omics experiment without a lot of hand holding, right? Very simple approach. Hope that helps.
Yeah, that's great. Thanks. Okay, last question, then we'll wrap things up here.
Sure.
Just getting back to the Saitonorm implementation.
Yeah.
So there was a question about the control sample, and if the control sample had, let's say, low expression in a certain marker, does that impact its ability to do the normalization? Or is there some kind of threshold in terms of expression level to be able to use it as a normalizing?
It's an interesting question. I can't say I will have a definitive answer for that. You do want to use control samples, like if you're stimulating the cells, you want to use the stimulated controls so that they have expression of the cytokines and the phosphoepitopes and such that you're quantifying. In terms of which control sample do you use? Well, as a human infectious disease immunologist, well, my favorite patient's right here, right on my own arm, but I don't have HIV. So if I want an HIV infected person, I might take a Buffy coat from one of my subjects in this patient cohort and freeze down 2 boxes of vials that have a phenotype, with cells that have a phenotype similar to the ones that I'm going to be querying in my experimental approach. And then each time I run a batch or an experiment, I can use that same exact specimen, and I know that that specimen from prior exploration has expression of the markers that I'm interested in. And that's an ideal situation where I have the ability to freeze those cells and make a control that is the same. It's much more difficult when you're using mice that you're sacking and you have, what control do you use there? Well, you might end up taking some splenocytes and doing the exact same thing, that you freeze them down so you can pull the same splenocyte population and use that as your control. As you do want it to match the real experimental samples as much as you can, but you have to work within the limitations of your experiment, what you have in terms of reagents and samples at your disposal.
Great. Yeah, no, that makes sense. Okay, well, yeah, I think we've gone on long enough, so we'll
2 hours,
go ahead and,
pushing 2 hours.
Yeah, not bad.
Thank you guys for the invite, and I'm sorry I go over. I spent my Easter running variations of UMap
Nice.
and putting together some of these new slides, and so I'm sorry it took a lot longer than some of my my other talks
that's
that
right.
did in the past, but hopefully it was more informative, and you know a little bit more about the options and how to implement this approach to discovery within the FlowJo platform than you did 2 hours ago. And again, as people have questions, I'm here for you guys. My tech support team here for you guys, and we'll be to discuss when you have it.
Great. Thank you so much, Tim.
Thank you.
Thank you all for joining us. Again, just to reiterate, ChUGcytometry.com. Join up on the forum and be happy to field questions there. If you need to revisit this live stream, you can check out that YouTube link or just go back to ChUGcytometry.com to follow along and revisit this broadcast. So on behalf of the newly minted ChUG group, thank you. Thanks to Laura. Thanks for David for helping setting this up and getting things going. The rest of the executive steering committee of ChUG, we look forward to seeing you online. And again, everybody stay safe. Hope you're enjoying your shelter in place amongst this SARS-CoV-2 epidemic. And hopefully we can all get back to work soon and test out a lot of these algorithms on newly created data as opposed to old OMIPs and things like that.
I will just remind you all that if you're stuck at home under quarantine, FlowJo is offering quarantine licenses so you can spin up a free 30-day license and do your analysis on your own workstation at home if you need it.
Great, thank you. Okay, with that, we'll sign off. Stay safe, everyone. Thanks for joining us and take care.